So previously we have talked about PCR amplification, and in PCR amplification we were actually talking about 3 different steps. The procedure was using 3 different steps in amplifying DNA or RNA. So the first step was the denaturation step, and then we have to lower down the temperature, and then the annealing step, and then we have again to raise the temperature, and then was the extension temperature. So this temperature was the denaturation temperature was 94 degrees, and this temperature, the annealing temperature was I'm just saying, for example, that is 55 degrees, and that was 72 degrees. So these 3 different temperatures we were using in the PCR. So that means the PCR has been conducted at 3 different temperatures. So this very first temperature has been divided into 2 parts, and this has been divided into 2 parts, and up till from here to here, this was actually we need to have 30 cycles or 35 cycles or 25 cycles depending upon the conditions and the requirements. So if we need 30 cycles, so that will mean that and this was initially for 10 minutes, and this was for 1 minute, and after this was like for 1 minute, and this was for 1 minute, and this was like for 10 minutes. So now conclusively, what we do actually in the PCR, first we need to have the initial denaturation for 5 or 10 minutes, and after that, this is actually 5 minutes, and that's range about 6 or 7, and this is for 1 minute, and then we need to lower down the temperature at 55 degrees so that the primer could anneal with the template, and after that for 1 minute, and then after that we have to raise the temperature up to 72 degrees centigrade for 1 minute, and then we have to have this temperature. After 72, we need to raise the temperature to 94, and then again the thing is same. We need to lower the temperature to 55, and then to raise the temperature to 72, and the cycles continues up and at a stage that the 30 cycles are complete, and then we need to have 10 minutes for final extension temperature. And then again, we need to lower the temperature and up till 4 degrees centigrade. So now if you can see, we need to have different temperatures, 94, 55, 72, and again 4. So all these temperatures, for changing these temperatures, we need a machine. We need an automated system, and that is Thermocycler. So we have thermocyclers. So that Thermocycler. So Thermocycler are PCR machines simply. What that works, that only change the temperature for us from 94 to 55 to 72 to 4. So these two different temperatures. So and this time from one temperature to the next temperature, this time the time that it takes is known as ramp temperature. So this ramp temperature is more important. This will be the ramp temperature. This will be the ramp temperature. So the less the time, the more efficient the thermocycler is, the PCR machine is. So that will mean this time the ramp time is important. And after that, so these different temperatures, for these different temperatures, we need a thermocycler, and the efficient thermocycler, the more good. The more good if we use a thermocycler, an efficient thermocycler. But if we want to skip this thing, and if we don't want to have different temperatures, either we can do all these stuff at a single temperature. Either it can be possible if we can do it at a single temperature, like at one temperature. And either it can be possible or not. Like if we can do that at 60 degrees, like if I want to proceed in PCR at 60 degrees. So I don't need I don't need any PCR machine. Then I just want I want to have a water bath, and I will maintain the temperature at 60 degrees, and the PCR amplification will occur at the same temperature. I don't want to have different temperatures, and I don't want to use equipment for my PCR. And that is possible with this technology, with isothermal amplification technology. So this is what we will be talking about in this topic, in isothermal amplification. Any question up till now, or everything is clear?
Clear, sir.
ठीक है. Okay. So isothermal amplification, what does it mean? It means that amplification or multiplication of the DNA at a very fixed temperature. And this will mean the detection of nucleic acid target sequence in a streamlined exponential manner. So the PCR will be streamlined, will be in exponential manner, just like in the PCR, but it will be more efficient than the PCR even. And then is not limited by the constraint of thermal cycling. We don't need to cycle, we don't need to have different temperature and the cycling of those temperatures. We don't need those cycling of temperatures. So after that, DNA strands are not heat denatured in isothermal amplification. We don't need any denaturation. Like in PCR, we need to have 94 degrees for heat denaturation. But here, we don't need any denaturation. And all isothermal amplification rely on an alternative approach to enable primer binding, how the primers will bind and how the initiation of amplification reaction will occur. And for this purpose, only a polymerase with displacement activity, we need only that thing. So bottom line is we will need a polymerase that will displace the strand, that will move the strand, and it will amplify the, it will bind, it will make the primer bind here at their location and will displace the strand to move amplification going on. Okay, unique polymerases for separated duplex DNA. We have different polymerases, and those polymerases first type, the type first in those polymerases is Clino-Exo, BSU large fragment, and 529. So these are three different types of polymerases that will work at moderate temperatures, like from 25 to 40 degrees. So if we choose one of these polymerases, like 529, or BSU large fragment polymerase, or Clino-Exo polymerase, so we need to amplify all the DNA at 25 or 30 or 40 degrees. So, but if we choose some other types of polymerases, like large fragment of BST DNA polymerase, we need to rise the temperature from 50 to 65 degrees. So we need to have 50 degrees temperature, or 60 degrees or 65 degrees, that depends. So we need to have higher temperature, but that will also be isothermal, that at one temperature the whole of the whole of the amplification will occur. And to detect RNA, if we have to detect RNA just like in the PCR, add a reverse transcriptase, and that should be compatible with the temperature. Like the reverse transcriptase, if that is working at 40 degrees centigrade, so we need to choose this polymerase so that the both reverse transcription and the amplification could occur at the same temperature. So it's compatible with the temperature of the reaction is added, except for NASBA and TMA reaction. We will be talking about this thing later on, to maintain the isothermal nature of the amplification. So we need just one thing we should be taking care of, is to take the reverse transcriptase that could be compatible with these polymerases. Actually. Okay, and these are certain types of technologies that are important, and we will be talking about these. The first one is loop mediated isothermal amplification. Simply, we will call it LAMP. That is loop mediated isothermal, and AMP for LAMP. So this is the L is for loop and the AMP for amplification. So loop mediated isothermal amplification. So first of the technologies is LAMP, and this is too much famous of these technologies, and this will be isothermal amplification. And the next is whole genome amplification, WGA, and multiple displacement amplification. That will mean we want multiple primers. We will use multiple primers to displace the template. And next is helicase dependent amplification. Here we will use helicase. That will the helicase will do. The helicase will actually will denature the double-stranded DNA, like we do. We need a high temperature in PCR. So here we will use helicase to do work to do the same work for us. And next is rolling circle amplification. Rolling circle amplification is like we have circular DNA and we want to amplify the and the circular DNA. So in that case, we will be using rolling circle amplification. And this rolling circle amplification, this is used for plasmid in nature. And the next of the technology, isothermal amplification technologies, is recombinase polymerase amplification. So RPA, and we will be talking about this in the slides later on. And next is nucleic acid sequence-based amplification, NASBA, and this will be we will talking about this in detail. So now NASBA, this amplification is used for RNA. Actually, if we have RNA and we want to produce cDNA, so we can use this technology, NASBA technology, for amplification of RNA actually. And if we talk about LAMP, we talked about this in detail previously. Is a single tube technique for amplification of DNA. It uses 4 to 6 primers, which form a loop-like structure to facilitate subsequent rounds of amplification. And here a point to note is we will need 4 to 6 primers, and means 4 is compulsory, and we can we have option of 2 more primers to use to increase the efficiency. And it will produce loop-like structures, and we will be talking about this in detail. And second is NASBA is a method to amplify RNA. So this will amplify RNA. If we need to amplify RNA, we will use NASBA technique. And third is MDA, and this is a technique that initiate multiple random primers need to DNA template and polymerase amplify DNA at constant temperature. So this will be multiple displacement amplification. We will be needing multiple random primers, and we will be talking about these random primers in detail. Don't you worry for this. So we will be talking about. So first we need to go through all the technologies first. And the next of the technology is WGA. When MGA is used to amplify DNA from a whole genome of cell, it is called a whole genome amplification. And some of the methods is of whole genome amplification. There's MALBSC, and that is multiple annealing and looping based amplification cycles. We will be talking about this. And LIANTI, and that is the linear amplification via transposon insertion. So this will be using transposon technology, actually, how the transposon is working, and we will be talking about this in detail. And next is DOPPCR, and that is the degenerate oligonucleotide prime PCR. Actually, the primers in this case will be degenerate, and we will be talking about the degeneracy in the primers later on. And the PTA, actually, this is the primary template directed amplification. Each of these technologies will be discussed later on. And next is HGA, and that is use of double-stranded DNA unwinding activity of helicase. Means this will be using helicase to unwind the DNA for us to separate the strands for in vitro DNA amplification at a constant temperature. And next is RPA, and that is a low temperature DNA RNA amplification technique using recombinase polymerase. So if we use a polymerase that has activity of recombination. So that will be that can work for us to amplify DNA and RNA. And RCA, and this simply is rolling circle amplification, and this starts from, it will require circular DNA as a template, and if the template is circular, we can use RCA for that, and a short DNA or RNA prime to form long single-stranded molecule. So in this case, in RCA, we will be needing circular DNA template. This is a bit different from the other ones. This will be the polymerase will have the polymerase activity. Here we will be using helicase. Here we will be amplifying the whole genome, and this is a type of this thing. And NASBA is used to amplify RNA, and LAMP is actually it will produce loop-like structure, and that's it. Anybody could explain? Who could explain? Sabah, Asif. Sabah? Can you hear me, Sabah? जी बताएं अब अभी तक जो पढ़ा है. आयशा अनवर, आप बताएं प्लीज.
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
ये बड़ा मुश्किल हो गया ना वैसे आयशा.
क्या सर?
हाँ अब ठीक है, ठीक है. वेट
Yes,
वेट
sir.
फॉर अ व्हाइल. मैं आवाज लाउड कर लूं. जी बताएं अभी तक क्या पढ़ा है. ये स्लाइड जस्ट बता दें थोड़ा सा. तो
सर इसमें ये है कि जो लैम्प है वो सिंगल ट्यूब में हम करते हैं. एम्प्लीफिकेशन होती है डीएनए की. 4 से 6 प्राइमर्स हम लोग यूज करते हैं और एक लूप लाइक स्ट्रक्चर्स बनता है, और उससे हमारे जो है मल्टीपल राउंड्स ऑफ एम्प्लीफिकेशन होती है. नासबा में जो है वो एम्पलीफाई करते हैं हम लोग आरएनए को. एमडीए जो है उसमें हम लोग मल्टीपल रैंडम प्राइमर्स यूज करते हैं और अपने टेम्पलेट डीएनए को सिंगल कांस्टेंट टेम्परेचर के ऊपर जो है वो एम्पलीफाई कर लेते हैं. और WGA भी है, जब हम MDA को यूज करते हैं होल जीनोम को एम्पलीफाई करने के लिए, तो उसके लिए तो वो WG है, उसमें मैलबैक और ये डॉप PCR PTA भी आता है. फिर HDA है जिसको हम यूज करते हैं डबल स्ट्रैंडेड DNA के लिए, और इसके पास हेलिकेस की एक्टिविटी है कि ये डबल स्ट्रैंडेड DNA को जो है वो अनवाइंड कर सकता है, और टेम्परेचर तो इनमें कांस्टेंट ही होता है, सिंगल होता है. और RPA जो है जब हमने लो टेम्परेचर के ऊपर एम्प्लीफिकेशन करनी हो DNA या RNA की, तो उसके लिए हम लोग जो है वो ये यूज करते हैं. साथ टेक्निक यूजिंग रिकम्बिनेस पॉलीमरेजेस, मतलब रिकम्बिनेस पॉलीमरेज एम्प्लीफिकेशन है ये. और दूसरी RCA रोलिंग सर्कुलर एम्प्लीफिकेशन है, जिसमें हम टेम्पलेट DNA जो है वो जिसको एम्पलीफाई करते हैं वो सर्कुलर फॉर्म में जो है वो एम्पलीफाई होता है.
Exactly. ठीक है. And who will be the next to explain? Anam Azam है. Anam, you there?
जी
Anam Azam, जी बताएं अनम.
सर को क्या बताऊं?
इस स्लाइड के बारे में ये डिफरेंट टेक्नोलॉजीज जो हैं लैम्प उसकी आइसोथर्मल एम्प्लीफिकेशन की, इसके बारे में बता दें क्या है.
एक सेकंड सर, आपकी आवाज बहुत लो आ रही है.
कह रहा हूँ इस स्लाइड के बारे में बता दें.
सर
अभी आ
इसमें
रही
लैम्प
आवाज?
का जी, इसमें लैम्प का बताया हुआ है कि ये एक सिंगल ट्यूब टेक्निक है और ये DNA के एम्प्लीफिकेशन के लिए ये भी यूज होती है, और इसमें 4 to 6 प्राइमर्स यूज होते हैं. ठीक है, और ये लूप स्ट्रक्चर जो होते हैं फॉर्म करते हैं. NSTA जो है वो RNA को एम्पलीफाई करने के लिए मेथड यूज होता है. जो MDA है वो जो टेक्निक होती है वो इनिशिएट करती है मल्टीपल रैंडम प्राइमर्स को अनिल करने के लिए DNA टेम्पलेट के, और पॉलीमरेज को एम्पलीफाइज करती है एट कांस्टेंट टेम्परेचर पर. और इसमें आरपीए का जो है वो है कि लो टेम्परेचर पर डीएनए और आरएनए को एम्पलीफाई करता है। आरसीए का बताया कि सर्कुलर डीएनए टेम्पलेट और शॉर्ट डीएनए और आरएनए प्राइमर को क्या कहते हैं फॉर्म करता है लॉन्ग सिंगल स्टैंडर्ड मॉलिक्यूल में।
ठीक है, चलो, we should go further. And the first of the technology is LAMP. This is LAMP is loop mediated amplification. I will never like explain the terms again, because here we have explained everything, and we will be just using the abbreviation later on. Okay, so LAMP is like, if you can see, so I think we have a video, and okay, so listen to the video, and we will be discussing the video in detail.
When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand-displacing DNA polymerase extends the primer and separates the target DNA duplex. When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand-displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure. When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand-displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms a seed for exponential LAMP amplification. This LAMP dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatenamers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.
Got it? Who will explain? Who will explain? Kuratulain is there. Kuratulain, sir? Can you hear me? Imran Liaquat, Imran? जी इमरान।
यस सर मेरा आवाज़ आ रही है, नेट का इशू आ रहा था जी सर।
क्या जी बताएं क्या?
सर क्या कह रहे हैं आप, काइंडली रिपीट कीजिए।
अच्छा, ये वीडियो में क्या देखा था तो थोड़ा सा एक्सप्लेन कर दें, हाउ इट इज डिफरेंट।
सर डिफरेंट ये है कि उसमें जो लैम्प के मुतालिक हमारे पास बताया गया कि जो लूप मेडिएटेड स्ट्रक्चर फॉर्मेशन है वो किस तरह इनिशिएट होती है, और 3 to 5 जो एंड है वहां से उसका जो है इनिशिएशन होती है और वहां पे लूप बनते जाते हैं जैसे जैसे न्यू स्ट्रैंड्स आते जाते हैं। यस सर।
तो हाउ कैन वन डिजाइन प्राइमर्स, प्राइमर किस तरह बना सकते हैं इसके लिए?
सर वो मेडिएटेड है ना कि हमारे पास पोजीशन आ रही है, वहां से हम आगे फर्दर अनिलिंग होती जा रही है और फर्दर हम बनाई जाएंगे जो हमारे लिए हेल्पफुल है।
और जो दूसरा नया स्ट्रैंड बनता है वो किस तरह रिमूव होगा, फिर डिस्प्लेस किस तरह होगा?
सर वो जो लूप स्ट्रक्चर बनता जा रहा है, जो ओल्ड वन है वो लूप में जाता जा रहा है और जो न्यू वन है सर वो स्कर्ल में होता जा रहा है।
वो
और
लूप में क्यों
जैसे
जाएगा?
सर वो लूप में सर वो उन्होंने स्ट्रक्चर में दिखाया है जो वीडियो में भी आपने दिखाया है।
चले अब देखें, एक्चुअली करते क्या है, लाइक हमारे पास हमने फॉर इंस्टेंस हमने हमारे पास डीएनए का एक सीक्वेंस है और हमने लैम्प पीसीआर लगाना है। सो फर्स्ट ऑफ द थिंग इज वी नीड टू आइडेंटिफाई द लोकेशन ऑफ प्राइमर्स, या हम प्राइमर डिजाइन करने। तो प्राइमर्स अगर हमने इस तरह डिजाइन करने, देख लें एक दफा, लाइक मैं डिजाइन कर रहा हूँ, हमारे पास एक सीक्वेंस है टी टी टी, और उसके बाद ये सीक्वेंस चलता जा रहा है और दूसरी जगह पे है लाइक दैट इज सी सी सी, वैसे मैं एग्जांपल के तौर पे वो कर रहा हूँ, एंड दैट इज जी जी जी। और this sequence goes on and I have like this sequence. अब होगा क्या कि ये सीक्वेंस मेरे पास है, सो मैं करूंगा क्या, सबसे पहले यहाँ के अगेंस्ट मैं प्राइमर डिजाइन करूंगा, एंड द प्राइमर विल बी लाइक प्राइमर विल बी जी जी जी, और ये जी 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 प्राइमर जो है ये इस तरह होगा, इट विल बाइंड हियर, और इसके साथ इस साइड पे चले। Okay, like if we have a sequence like T T T, and then again like C G C, like this sequence we have G G C, and we have this sequence, and we need to design the forward primer first. So one of the primer will be like, I will be having a primer, and that will be GGG, and this GGG will be bound with okay, and this will be bound with a sequence which will not bind there, and this will be like the same sequence, like this is complementary to this sequence. This sequence will be complementary, and here I will bind it with another sequence, and that sequence is exactly the same sequence here, and I will bind it with the, so the primer will be like this, GGG, GGC, and this will, so look at this, this will bind here, this will bind here, and this will bind here. Now this sequence cannot bind there because this is the same sequence. Now when the amplification occur, what will happen? It will be like sequences, and after that amplification. When amplification occurs, it will produce like C C G, and then this will be like this. So after production of this, the new strand, what will happen? Now we need to separate the new strand, and we need, because this will bind here, here, and here. So we need to remove the newly formed strand from this. So this is the first of the objective that we need to remove. So we need another primer, another primer, and that primer will be here. In this case, will be AAA. So this primer will bind with here, and this will start amplification in this direction. So this will start amplification in this direction, and this will remove this strand, and this will move down, and how it will form a loop-like structure, like it will form, and this newly formed strand will be like this, like CCG, and so on. And before that, here this strand, this G, this will be GGC, and this will bind with here, this is this thing, and here we have GGG. So it will be like this. It will bind like here and here, and it will bind like this one. These three bonds will occur there. So this will form a loop-like structure. So this is the first of the loop, and to produce a dumbbell-like structure, we need to use the reverse primers as well. By using reverse primers, we will be having a dumbbell-like structure like this, and it will be like this. So this dumbbell structure, after production of this dumbbell structure, this is the first step that it will be the loop mediated amplification will be started first. Here these primer will be used, the same primers will be used. This will amplify in this direction. Similarly, if this is 3' end and this is the 5' end, so it will, the 3' end will serve as the amplification start point. So it will start amplification, and now it will be the DNA will be like this. This end will be like this thing, and this will be like this. So this will increase, and it will be like this structure. So and when the other primer is used, so it will produce, it will amplify this strand, and this will the concatenamers will occur, will form by, and now this primer, and when the primer attach here, and it will produce the whole amplificon, and the concatenamers will increase in number. It will, like if we have a unit length of 1 kb, so there will be amplicons that will be having 1 kb. Some of the amplicons will be having 2 kb in length. Some will be having 3 kb, some will be having the length of 3 kbs. So these are concatenamers. So these are, this will be trimer, this 1mer, 2mer, 3mer of the whole length. So this will be trimer, tetramer, and so on. So these multimeric structures will form during the loop amplification. So one more thing, this loop, the advantages of loop is assured. This is actually the WHO recommendation, and that is affordable, sensitive, specific, user-friendly, robust and rapid, equipment-free, and deliverable to the end user. So this applies to the lamp as well. And next of the technology, okay, so why lamp, why the name occurred? Because it is loop mediated. It will produce loop-like structures, and the loop-like structures are formed when lamp primers amplify the target DNA. And next is it will work at a constant temperature, a constant fixed temperature, and therefore it is isothermal. So, and next is amplification. So amplification will be efficient, and they can detect very small amount of target DNA. and so this means this is very sensitive, because if there is a very low amount of the target DNA, that can be amplified, which is not possible with the conventional PCR. And for the LAMP primers, it will be having forward inner primer, and this will be hybrid primer. We have discussed that thing, that will be hybrid, and backward inner primer. So forward inner primer, backward inner primer, forward outer primer. This outer primer is required to displace the newly formed strand from the template. So the strand and the strand and the template hybrid. So that will be removed by using forward or backward outer primer. The outer primers are required actually. And the forward loop primer and forward backward loop primer, they are used to enhance the efficiency. We will be talking about this in the coming slide actually. And forward loop and backward loop, this actually accelerates the amplification reaction significantly. And if we don't use forward and backward primer, loop primer, don't you worry. We have the 4 primers that could do our work for us. And this is actually the schematic diagram. Like this is the F3 position, F2 position, and F1 position, and similarly B1 position, B2, and the backward 3 position. And this is actually the complementary sequence of this strand. So F3C, this is the complementary, F2C complementary, and F1 complementary sequence, and the same is with this case. Okay, so now another region is a forward loop primer. This is the loop-like structure that will be formed when the loop-like structure will occur, and this region, the primer can be used for this region as well. And same is the case with backward loop primer. So how we can design, actually, if we have this DNA, so how can we design the forward and backward primers? So if we want to target this DNA, the F2C, so we will take this F2, this will bind here because this F2 is complementary to F2C. It will bind here, and if we will be using F1C because that the newly strand form will be having F1. And F1C will be complementary to F1, the newly formed strand, and that will form a loop-like structure. And similarly, we will be using, actually we have used a complementary strand, a complementary primer to this strand. So now the outer primer will be complementary to this thing. So F3, for F3, we will be using the primer that, so this will bind with F3C, and this will displace the newly formed strand, actually, this forward primer. And forward loop primer, that is enhance the amplification. Similarly is the backward internal primer. So for backward internal primer, that will be binding with that will be binding with this B2 because that is B2C. When this binds this, it will amplify B1C, and when B1C is formed, sorry, B2, this will bind with the B2. This will bind with B2, and it will produce B1C actually, and B1 will bind with B1C, this B1. And now it will form a loop-like structure when it is displaced from the template strand by using the backward outer primer. Then the backward outer primer is this B3, and this B3 will bind here, and it will displace the newly formed strand actually. And same is the case with the backward loop primer. So backward loop primer will bind here, and it will amplify. It will enhance the amplification. So this is another representation of the same procedure. We have double-stranded DNA, and this double-stranded DNA is first amplified with the first set of primer, and that is F1 forward internal primer, and that will bind with this internal region. And when it binds here, it will amplify, and this will amplify this F1 and one is this F1. So the F1C and F1 will be complementary, but the whole strand will not be removed until and unless we use an outer primer, forward outer primer, and this outer primer, the black one, this will amplify the region, and this will displace the newly formed strand. And now you can see the newly formed strand has been displaced, and this F3, the outer primer, is now has amplified the sequence in that sense. Now when this strand is amplified, this will produce a loop-like structure. Now the turn for the backward primer. So backward internal primer will amplify the sequence. When it's amplified, the amplification is complete. The outer internal primer will be there. That will the backward outer primer. So that will bind here, and that will amplify the sequence by displacing the newly formed sequence. And this newly formed sequence will now be a dumbbell structure. Now this dumbbell structure will be the starting point for loop amplification. So you can see this structure will produce the concatenamers, and these concatenamers will either produce monomers, or they will increase in the concatemerization. It will be more of the subunits will be produced, and this will be from dimer to tetramer to hexamer and so on. So this is how this goes exponentially. So in one reaction set, we will be having different amplicons, and this will amplify in exponential manner. So the primer that will bind here now can also bind here as well, and the 3' end of each of the template will be serving as a starting point to initiate the replication. So it will increase in the length actually. So I think we have another video. Okay, so no. So a lamp amplification. So how we can detect how we can detect either the amplification has been occurred or not. So there are different techniques. For normal conventional PCR, we were using Agarose Gel Electrophoresis, but here, as the amplification occurs exponentially and more rapid, we have different techniques for that. And the first of that is by using magnesium precipitation. So we already know that inorganic phosphates accumulates in the reaction. How it occurs, how the inorganic pyrophosphate accumulates by adding a nucleotide. The pyrophosphates actually release the reaction actually, and we get pyrophosphates, inorganic phosphates. So those pyrophosphates can react with magnesium to produce magnesium pyrophosphate, and that will precipitate out, and this will be a visual indicator. समझ आ रही है? So अगर precipitation आ जाए, so that will mean कि reaction is in process. Can you hear me? जी बताएं.
सर ये जो पीछे आपने डायग्राम दिखाई है, हम लोग ने पहले भी देखा है कि हम लोग जो है सेकंड स्टेप से क्यों स्टार्ट करते हैं, हम फर्स्ट बेस क्यों छोड़ देते हैं हमेशा.
मैं आपकी बात नहीं समझा, रिपीट करें.
सर पीछे डायग्राम दिखाएं.
This one, yes.
जी ये देखिए ना, हमने F3C को छोड़ दिया और F2C में हम आ गए, ठीक है. और जो हमने फॉरवर्ड इनर प्राइमर लगाया है वो हमने F2C से स्टार्ट किया, ठीक है. हमने F3C क्यों छोड़ा है हर जगह पे.
ओके, आप ये कह रही हैं कि हम F2 से क्यों स्टार्ट कर रहे हैं, F3 से क्यों स्टार्ट ना करें कि एंड से ही स्टार्ट हो.
हाँ जी.
अच्छा, सो ये देखें, ये बड़ा ट्रिकी पॉइंट है. अब देखें, अगर हम F3 से स्टार्ट करें, ठीक है, सिंपल इज दिस. अगर यहां से स्टार्ट करें, सो जो न्यूली फॉर्म्ड एम्प्लिकॉन है ना, ये जब न्यू स्ट्रैंड बन जाएगा, सो ये यहां से सेपरेट किस तरह होगा. वी विल बी नीडिंग हाई टेम्परेचर. हमें 94 डिग्री सेंटीग्रेड टेम्परेचर इसको देना पड़ेगा।
सही।
ठीक है। सो उस टेम्परेचर से बचने के लिए अगर हम यहाँ पे इसको ये वाला प्राइमर ले लें और आउटर प्राइमर जब यहाँ पे बाइंड करेगा तो ये दूसरा जो न्यूली फॉर्म ये बनेगा ना, तो ये इसको डिस्प्लेस करता जाएगा और ये अलहदा हो जाएगा, ये दिस वन, एंड दिस विल बाइंड देयर।
लेकिन हम इनिशिएट हम उसको हाई टेम्परेचर से करेंगे, फिर हम F2C की तरफ आएंगे, ऐसे ही है ना सर?
इनिशिएट कहां से करेंगे?
हाई टेम्परेचर से।
हाँ, इसमें कुछ टेक्नोलॉजीज हैं जिसमें हाई टेम्परेचर से इनिशिएट करेंगे, लेकिन पॉलीमरेज ने क्या करना, इट हैज स्ट्रैंड डिस्प्लेसमेंट। ये पहले जो इंसर्जन है ये पॉलीमरेज ने करनी है। वी डोंट नीड हाई टेम्परेचर।
सही हो गया।
कुछ केसेस में हमें हाई टेम्परेचर की जरूरत होती है, वो आगे जाके कुछ टेक्नोलॉजीज इस तरह है कि जिसमें पहले हम आइसोथर्मल एम्प्लीफिकेशन करते हैं और फिर हाई टेम्परेचर पे जाते हैं। कुछ हैं टेक्नोलॉजीज इस तरह। ठीक है। सो बट फॉर नाउ दिस विल बी एट सेम टेम्परेचर। ठीक है।
ओके सर, ठीक हो गया।
चलो, एंड
दैट वाज कंफ्यूजिंग फॉर मी।
सो ये बताएं इसके बारे में, ये मैग्नीशियम क्लोराइड के बारे में, मैग्नीशियम के बारे में, इसमें क्या होता है? नूरुलैन बताएं।
जिससे मैं अभी पिछले पॉइंट में फंसी हुई थी।
अच्छा, उससे फंसी हुई थी। ठीक है, चलें चलें। अक्सा यहिया। चलें, सबा सरफराज। जी, ये मैग्नीशियम प्रेसिपिटेशन क्या है?
यस सर।
जी, मैग्नीशियम प्रेसिपिटेशन क्या है ये?
सर ये बताया कि जी इनऑर्गेनिक जो पायरोफॉस्फेट्स हैं वो जो एक्यूमुलेट हो रहे हैं ना, उनकी प्रेसिपिटेशन हो रही है, उससे मैग्नीशियम पार्टिसिपेशन कह रहे हैं। और रिएक्ट्स विद मैग्नीशियम आयन टू प्रोड्यूस मैग्नीशियम पायरोफॉस्फेट।
नहीं, तो इसने क्या करना है पायरोफॉस्फेट ने?
जब उनके साथ रिएक्ट करना है तो उससे वेसल में से इंडिकेट करना है।
प्रेसिपिटेशन बनेंगे, प्रेसिपिटेट्स, व्हाट विल बी प्रेसिपिटेटिंग, व्हाई वी नीड दैट?
करेंगे।
हाँ,
इंडिकेशन के लिए यूज कर रहे हैं ना ये मैग्नीशियम।
हाँ, that will be a visual indicator। ठीक है। और पायरोफॉस्फेट्स हम पहले कहाँ पढ़ चुके हैं वैसे?
सर सीक्वेंसिंग में पायरोफॉस्फेट सीक्वेंसिंग।
यस सर, सीक्वेंसिंग।
So this is another application for that, no? Okay, pyrophosphates are producing, and we will be adding magnesium, and this will precipitate the pyrophosphates out, and this will be a visual indicator. We can identify either the reaction is occurring or not, either the reaction is in process or not.
जी sir.
Okay, and next is colorimetric technique of detection of the lamp amplification. So we already have talked about the ion torrent, and in that we have learned that by addition of one nucleotide, the pH decreased by one hydrogen ion will be added to the solution. So one proton actually. So that will change the pH. So if we use different dyes like that, the dyes that are sensitive to the pH, so that will give us different colors, and with that color we can determine either the amplification is in process or not. Am I clear?
Yes sir.
Yes sir.
Okay, and the next is, you already know, the Agarose Gel Electrophoresis. But for now, the Agarose Gel Electrophoresis, we will not be able to see a sharp single band in the Agarose Gel Electrophoresis. because we will be having concatenamers, and the concatenamers will be either monomer, dimer, trimer, and so on. So we will be having a ladder-like structure. So we will not have a single band actually. But if we use some restriction digestion enzyme, if we could use some enzyme, we can have a single band in this as well. You got my point or not? मेरी बात की समझ आ गई है कि नहीं?
नो
जी
सर।
सर, यार वो मिक्स पीक्स आएंगे ना।
किसको समझ आई है वो बताएं।
यार मैं बताऊं?
जी जी बताएं नूर।
सर इसमें ये होता है कि हमारे पास जो है ना मिक्स्ड पीक्स आती हैं, हमारे पास इतने ज्यादा मतलब, और रिजल्ट्स जो होते हैं ना वो ओवरलैपिंग रिजल्ट्स आते हैं इसमें, तो हम लोग डिस्टिंग्विश नहीं कर सकते होते हैं कि हमारे पास क्या आया, मतलब हमारा जो फाइनल रिजल्ट है वो क्लियर कट नहीं होता इसमें।
कोई और बता रहे? अच्छा आयशा अनवर आप बताएं।
सर मैंने ही तो पूछा, मुझे समझ ही नहीं आई कि वो जब कॉन्केटेमर्स बन रहे हैं, मोनोमर, डाइमर, ट्राइमर है, तो आपने कहा कि हम रिस्ट्रिक्शन डाइजेशन करते हैं, तो मतलब वो जो एंजाइम्स होंगे वो क्या मोनोमर्स की फॉर्म में उनको कट कर देंगे?
इस पॉइंट को समझ ले, दिस इज इम्पोर्टेंट। सो लाइक हमारे पास इस तरह डिफरेंट लेंथ के डीएनए आ जाएंगे, डबल स्ट्रैंडेड डीएनए आ जाएंगे डिफरेंट लेंथ्स के, ठीक है, कुछ ज्यादा होंगे, कुछ लेंथ में ज्यादा होंगे, कुछ कम होंगे, डिफरेंट लेंथ के डबल स्ट्रैंडेड डीएनए आ जाएंगे, ठीक है। सो हाउ कुड वी नो के लाइक what is the size of the DNA? Because if we run these DNA on the gel, they will get we will get different size bands. So what will happen if we use if we can say this is 1 unit length, this will be 2 unit lengths, this is 1, this is 2, this will be 3, and this will be 4. Like we have 1, 2, we have like a sequence of 10 kb. So this will be 10 kb, this will be 10 kb, and this will be the repeat of that 10 kb, and this will be the repeat of that 10 kb, and this again. Okay, so now if you can see, this will be here, like this will be here, and this will be here. So if you can see, this is a tetramer, this will be a trimer, this will be a dimer, like I say it like this. This will be a dimer, this will be also a dimer, and this will be a monomer. So sorry, I have to make it like this. So this will be like dimer. Okay, so we have different size of the amplicons. So what we can do, how we could know either the same amplification has been occurred or not, the same size of the amplification, we need same amplification. So if we know the sequence, so if this sequence has if this sequence has eco R1 here, eco R1 site, like I write it like here, this is E1 site, this is eco R1 site, this is like ECO R1. So this is eco R1 site here. So this site, well, that will mean this site is also here, this site is also here. Got my point? And this site will be also like here, because that is the same copy of the same. And this will be here, this will be here, the eco R1 site, and this will be here again, this will be here, and this will be here. So now, and this will be here, and this will be here, and that's it. So if I use eco R1 site, so try to understand, if I use eco R1 restriction enzyme, it will cut at the respective sites, and we will only get 1 unit length, and we will get only 1 band. समझ आ रही है? समझ आ रही है? ये सारे यहाँ पे जो रिस्ट्रिक्शन साइट्स है ना, ये कट जाएगा, यहाँ से ये कट जाएगा, तो ये देखें, एक यूनिट लेंथ ये है, ये एक यूनिट लेंथ ये है, एंड यहाँ से ये कट जाएगा, सो ये एक यूनिट लेंथ, ये सारे इकट्ठे होके एक ही बैंड बनेगा। समझ आ गई कि नहीं?
Yes, sir.
ठीक है, सो वी विल बी हैविंग ओनली वन बैंड इन देयर ऑन द जेल। समझ आ गई सबको कि नहीं? बताएं।
यस सर। सर
जी जी
सर, लेकिन आपने तो कहा कि ये जो एम्प्लीफिकेशन होती है इसमें हमारे पास मतलब वेरिंग लेंथ हो सकती है, कोई 2 kb का फ्रेगमेंट हो सकता है, कोई 3 kb का नहीं।
हाँ, वेरिंग लेंथ होंगे ना, ये देखें वेरिंग लेंथ है, ये इनिशियली वेरिंग लेंथ है। अगर हमने उसको जेल पे रन कर दिया तो वो हमारे पास एक बैंड नहीं आएगा। लेकिन अगर हमें किसी एंजाइम का पता हो कि कहीं पे वो कट कर रहा है, एक ही साइट है उसकी, सो अगर वो वहां से काटेगा तो सारे इक्वल लेंथ में आ जाएंगे। आप इसको ट्राई करके देखें, आपको खुद पता चल जाएगा कि सारे सेम साइज पे आ जाएंगे। लाइक सिंपल समझ लें, अगर मेरे पास ये एक लॉन्ग डीएनए है और और ये एक यूनिट है, ये इसका रिपीट है, ये इसका यहां से यहां तक, ये इसका यहां से यहां तक रिपीट है, और इसके बाद ये रिपीट। अगर एक साइट मुझे यहां पता है कि वो बिल्कुल दरमियान में है, ठीक है, ये साइट रिस्ट्रिक्शन साइट है यहाँ पे, ठीक है। अगर मैं ये करूंगा कि, और ये यहां से पहले भी अगर फॉर इंस्टेंस कोई है, सो अब क्या होगा कि जब मैं रिस्ट्रिक्शन डाइजेशन लगाऊंगा उसके बाद मेरे पास लेंथ क्या होगा, एक लेंथ इज दिस वन, क्योंकि ये यहां से कट जाएगा ना, तो मेरे पास ये फ्रेगमेंट आ जाना है। दूसरा फ्रेगमेंट यहां से शुरू होके यहां तक होना है, तीसरा फ्रेगमेंट यहां से शुरू होके यहां तक होना है, एंड सो ऑन। ठीक है, अब ये सारे जो हैं ये सेम लेंथ के हैं, ये वाला एंड दिस वन। ठीक है, जब मैं इसको डाइजेशन के बाद जेल पे रन करूंगा, आई विल गेट ओनली वन बैंड। ठीक है, मेरे पास सिर्फ एक बैंड आना है। ठीक है, उससे पहले जो है ये सारे इस तरह कंकैटिमर्स थे, डिफरेंट बैंड्स थे यहाँ पे, ये वाले, ये डिफरेंट बैंड से पहले। ठीक है, जो डाइजेशन के बाद सिर्फ एक बैंड आ गया। समझ आ गई है? स्टिल इफ यू हैव क्वेश्चन try to answer or try to ask actually.
सर अब आप अपना क्वेश्चन जरा रिपीट करें, समझ तो आ गई, क्वेश्चन क्या था?
नहीं, मैं कह रहा हूँ कि समझ आ गई अभी तक जो हो गया।
सर आपने क्वेश्चन जो किया था वो दोबारा बताएं।
अच्छा, क्वेश्चन ये था कि जेल पे जब रन करेंगे तो हमारे पास डिफरेंट बैंड्स आएंगे ना। जेल पे रन किया एंड हमारे पास डिफरेंट बैंड्स आ गए, इसकी तो समझ आती है ना? डिफरेंट बैंड्स आएंगे, लेकिन डाइजेशन के बाद सिंगल बैंड क्यों आएगा? ठीक है, समझ आ गई है?
जी सर अब मैंने नहीं आई।
चले दैट्स गुड। अच्छा, और किसको समझ नहीं आई? रेहाना शाहीन, रेहाना।
एक्सक्यूज मी सर।
जी
सर मुझे समझ आ
सर
रही है,
आपने
पहले नहीं आ रही थी।
ठीक है रेहाना, ठीक है। जी आप
सर आपने जो एक्सप्लेन किया है, जो अपने यहाँ पे अभी हमें शो किया है, ठीक है, वहां तक की तो समझ आ गई है। लेकिन सर इसमें जो इधर लाइन में जो लिखा हुआ है मोनोमर, ड्राइमर और ट्राइमर, उसकी समझ नहीं आई कि वो मोनोमर और डाइमर कैसे शो होंगे। आपने कहा था हमें एक ही बैंड शो होगा, लाइक 2-3 भी अगर हमारे पास फ्रेगमेंट्स आते हैं तो मतलब वो हमें एक शो होगा बैंड, लेकिन फिर सर उसमें मोनोमर डाइमर का कैसे पता चलेगा?
अच्छा, ये देखें यहाँ पे आपको नजर आ रहे हैं ना, ये मोनोमर है, ये एक यूनिट लेंथ है। ये देखें, ये यहाँ पे रिपीट, फिर ये रिपीट, फिर ये रिपीट, तो ये टेट्रामर है, और ये आगे बढ़ते जा रहे हैं। तो सोल्यूशन के अंदर ये सारे होंगे, मोनोमर भी होंगे, डायमर, टेट्रामर, और उससे लॉन्गर साइजेस के भी होंगे। ठीक है,
ओके सर।
सोल्यूशन के अंदर सारे होंगे। अब यहाँ पे देखें, अगर एक इको R1 की साइट यहाँ पे है, सो दैट विल कट हियर, सब में कट लग जाएगा, सिर्फ एक यूनिट लेंथ का आ जाना है हमारे पास। ठीक है,
ठीक है सर।
ठीक है, इस तरह सारा सब में इसी तरह होगा। एनी क्वेश्चन, एनी पॉइंट, एनी सजेशन? Okay, should we go further?
Yes, sir.
Okay, and electrophoresis. And these are some visual tests. And the first one is some, we can do real-time monitoring. We can also observe Agarose Gel Electrophoresis, and you can see a smear-like structure or ladder-like structure has been observed. And the third one is visual turbidity, like this will be turbid, the precipitates will occur. The next one is like if we use pH sensitive dyes, and that will change the color. And the next one is they use the under UV light, so they will fluorescence. They will have different fluorescence under UV light. So we have a video, and let's check it out, and then we will proceed further. We will talk about this as well.
There long has been a promise of isothermal and other amplification methods that don't require instruments to work in the field for immediate detection of pathogens or of parasites or things like that. But they've been limited a bit in real application. You had to wait a long time, or you had to observe at a very precise time in the detection. We wanted something a little more robust. So we took advantage of a natural product of polymerase reactions, the production of a proton that's used in some sequencing methods. So coupled that to an ensemble reaction and a natural pairing with a pH indicator to make a fast, robust visual detection. So here's just an example of our dyes in action. So at the top, just a strip tube of the start of a reaction where everything is the same color. Then down here we indicate positive reaction where amplification has taken place. And you see a nice change from the sort of clearish orange color to a bright pink. And you see a very clear discrimination of positive and negative reactions. Our detection method is very stable, so if you run the reactions, you can leave them on the bench for days at a time and check them at any time of your convenience. In our method, we use classical, traditional pH indicators for the visual detection, but the key feature to allow them to function for amplification was enzymes that can tolerate a wide range of pHs and perform without buffer. So using some of the novel isothermal reaction reagents from NEB really allowed these techniques to work. We're currently working with researchers in NEB's parasitology division to develop tests and methods for surveillance in the field of insect vectors and filarial parasites. We have a big interest in neglected tropical diseases, so we hope our method can really push these techniques to their limits and enable their true application in field and point-of-care settings.
So next is so we have another video, and this video don't have any sound, so check it out. And this is actually the overview of the whole LAMP procedure we have talked about earlier. And you can see we will be having, we will be using the inner primer, the loop primers as well. And this 3' end will work as a primer start point. This will be the starting point for the amplification, and to amplify like this. Here the primer can bind, and a loop primer can also work here. And check it out, and we will discuss this. So this is the outer primer. Now this releases the newly formed strand. Now the time for the reverse primers, and now the time for the outer primer that would release the sequence. Yeah, this is releasing now the newly formed sequence. This is how the technology works now. Now the dumbbell structure occurred, and now the start of the reaction actually. The 3' end will start, and this primer can work, and the 3' end can work. Now the forward loop prime can work, And these are different types of the products that will occur in the same reaction actually. And next of it is whole genome amplification. अभी तक समझ आ गई सारी चीजों की class?
Yes, sir.
Okay, and now the whole, that was isothermal amplification, the lamp amplification. Now whole genome amplification. This is another type of isothermal amplification, and this is to increase the amount of limited DNA targets. Isothermal whole genome amplification is the most efficient technique. This is particularly useful in genetic research, disease research, where many repetition is required. So this is a highly efficient technology, and DNA amplified by WGA is used in downstream NGS, Sanger genotype with microarrays, SNP genotyping. So all of these they can employ the DNA amplification with whole genome amplification. So WGA is applied to all the NGS system platforms, the Sanger sequencing platform, genotyping for microarrays, and so on. So, and the most important is the 529 DNA polymerase, and this is the main enzyme of choice for whole genome amplification. And next is MDA, and I have already tell you that already talked about it, that MDA is a type of whole genome amplification, and this employ random hexamers. The random hexamers are used, and they will bind to the denatured DNA. So random hexamers are used instead of the specific primers. They will be primers but they will be random hexamers. I mean, they will consist of 6 nucleotides. 8 nucleotides. And this is followed by synthesis of DNA using 5 DNA polymerase. So DNA polymerase will do polymerization, and the primers will be used, hexamers will be used. And this polymerase does not easily dissociate and generates DNA up to 100 kbs. And this polymerase can also resolve secondary structure, including a hairpin loop structure. It says that what will happen if we use this, if we have a DNA strand and the primer binds here, and if we use 529 DNA polymerase, it will go till the end, then up to 10 kbs it will amplify, and we actually don't need that, this long amplification. Why we don't need them? Let's check it out in the video, and we will be talking about this later on. But it cannot easily dissociate. This is an important issue, and how this has been addressed. Let's check out the video. समझ आ गई कि नहीं कि why we want to go for the video? Because we want to know why, how the polymerase could be dissociated from the DNA strand.
Welcome to 3 Minutes with QIAGEN, where you will get answers to specific questions in 3 minutes. The products shown here are intended for molecular biology applications only. These products are not intended for the diagnosis, prevention, or treatment of a disease. QIAGEN offers complete solutions from sample to insight for performing next-generation sequencing from single cells. Each mammalian cell contains only 6 picograms of genomic DNA, while most NGS methods require nanograms of DNA. Hence, whole genome amplification is a must. Whole genome amplification can be done by PCR-based methods or non-PCR-based methods, such as MDA. The question today is, why is MDA the most preferred method of whole genome amplification?
During PCR, DNA templates undergo a number of cycles, including heating steps at 95 degrees Celsius for denaturation. Repeated exposure to high temperatures leads to DNA damage, deep urination, and strand breakage. This will result in amplification bias that will affect sequencing results. Multiple displacement amplification, MDA, is an isothermic amplification process where the amplification is carried out at 30 degrees Celsius. The low temperature helps to keep the tiny starting material intact and reduces DNA template damage. Secondly, MDA uses 529 DNA polymerase that has 3 important properties. Compared to Taq DNA polymerase, which is normally used in PCR-based methods, Phi29 DNA polymerase has a 1,000-fold higher fidelity, resulting in very low error rates. The Phi29 enzyme has very high affinity for the template, and this makes it suitable for use with a small amount of material. Phi29 also overcomes the challenges presented by the secondary structure of DNA in GC content, resulting in a highly uniform coverage of the genomic regions. Phi29 DNA polymerase also has extraordinary processing capability. Each highly processive Phi29 enzyme remains bound and continues replicating a single template for long stretches up to 1,000 kilobytes. This processivity also means there are fewer polymerase binding events and less opportunity for certain templates to become highly overrepresented. The combination of high template affinity and extraordinary processing capability enables it to generate long fragments.
How does QIAGEN apply this technology?
QIAGEN offers the Replication single-cell library kit, which is based on MDA technology. The kit uses a gentle alkaline incubation strategy, allowing effective cell lysis and highly uniform gDNA denaturation with low DNA fragmentation or generation of abasic sites, thus maintaining DNA integrity. The specially developed Phi29 variant leads to high yields of long amplitude fragments with high accuracy. The library preparation protocol is a simple 1-2 procedure that reduces the risk of losing DNA in the intermediate steps, resulting in high yields of superior quality libraries that do not need further PCR enrichment steps. Overall, the combination of MDA technology and Phi29 polymerase and the Replication single-cell library kit enables you to prepare sequencing libraries with high fidelity and accuracy, while retaining the sample genomic diversity in about 5.5 hours.
For additional information, please listen to our webinar series on single-cell analysis, Sample to Insight Overview.
So, commonly used WGA methods that are DOP, PCR, MDA, MALBSC, LIANTI have drawbacks. So, we have talked about MDA, and we will be talking about the other later on, but all WGA methods that have been given above, they have drawbacks, and the drawbacks are some of them have amplification bias, poor uniformity, errors, artifacts, low genome coverage, inability to address all variant classes, low accuracy, poor reproducibility, and complex protocols and difficult to automate or scale. So these are the drawbacks of these WGA methods. So how they could be how they could be overcome. Actually, one of the problems which I could like to explain is like it will amplify from this region more than this. Some of the amplification will be more from this region. Some of the amplification will be from this region. More of the amplification. So maybe these are few regions that are ignored during the amplification. So the whole genome amplification, they have problems like this, and therefore there will be amplification bias. The bias is towards the amplification. Some of the regions will be more like they will be suitable candidates for the amplification. The more amplification will occur from here, and they will be ignored due to some reasons. So how they could be overcome, and we will be talking about this, and they could be overcome by using PTA. And PTA is also a type of whole genome amplification. And this PTA is novel isothermal whole genome amplification method that reproducibly captures more than 95% of the genome in a single cell and in a controlled and more uniform and accurate manner than existing approaches. This improves variant calling sensitivity. This will be sensitive, specific, and will be economic, and they will be facilitating bioinformatic analysis. So this will be more feasible actually. And PTA take advantage of processivity, strand displacement. They employ actually the strand displacement activity and the low error rate of 529 DNA polymerase. So DNA polymerase has low error rates. And the PTA also employs exonuclease resistant terminators. So this is addition to the previous one. They will use terminators, and these terminators, the use of these terminators are too much important. We will be talking about later on in these slides actually. So the amplification products that are what these terminators will now do, they will create relatively short double-stranded amplification products that are poor templates for some subsequent cycles of the amplification. I have already talked about this. Like, we will be amplifying the sequence from the original template, and once the amplification has occurred, the amplification products will not be used for further amplification. And this transforms the reaction from an exponential into a quasi-linear process, and now this will be no more exponential amplification, during which majority amplicons are generated from the primary template. Okay, and unlike MDA, PTA, what PTA will do, and that will limit the exponential propagation of priming and amplification biases, allelic skewing, and other errors from the daughter molecule. This results into improved uniformity, coverage, and reproducibility, and significantly improved variant rates. How it will do? Let's check the video, and we will understand how it will occur.
Diversity is a fundamental part of life on Earth. We observe it every day in every aspect of our lives. Variation is also a natural feature of biological systems. Genetic variation has long been associated with complex disease states. In the case of cancer, genetic heterogeneity among cells is the norm, and accurately assessing the genomes of single cells is critical to individualized therapy. Primary template-directed amplification, or PTA, amplifies individual genomes in preparation for downstream genetic analysis in a way that offers unprecedented accuracy and uniformity. Similar to current methods, primers first anneal to the target genome, called a primary template. When DNA polymerase Phi29 binds to the primer and template, it synthesizes a complementary strand from the primary template. Chain-terminating nucleotides are then introduced, restricting amplicon length by disengaging the Phi29 enzyme and preventing further extension of the amplicon. The shorter amplicons have a significantly reduced propensity for reuse, replicating only a handful of times before becoming fully double-stranded. This preferentially directs the primers back towards the primary template of interest. Primary template-directed amplification provides researchers with an unprecedented level of cellular resolution, enabling a new era of genomic discoveries across a multitude of applications. PTA offers superior performance compared to traditional MDA. Redirection of primers to the primary template prevents the propagation of errors that is common with traditional MDA. PTA also offers significantly higher breadth of coverage and uniformity, resulting in increased sensitivity. With this increase in coverage uniformity, researchers observe far less allelic imbalance compared to MDA. Some examples of applications of PTA include cancer diagnosis and treatment, significantly higher breadth of coverage compared to.
जी बताएं क्या?
सर आपकी आवाज नहीं क्लियर आ रही, वीडियो की क्लियर आ रही है.
Okay, इसको दोबारा देखें. This point is too much important. ठीक है, यहाँ से कि ये पूरे क्रोमोसोम को कवर कर रहा है, और PTA और जो MDA है वो पार्ट्स ऑफ द क्रोमोसोम्स वो कवर कर रहा है, पूरे क्रोमोसोम को कवर नहीं कर रहा.
कर दें, सेलुलर रेजोल्यूशन से.
जी अब बताएं क्या बताएं.
सर इसको थोड़ा सा और प्रीवियस कर दें प्लीज.
ओके, फ्रॉम द स्टार्ट.
थैंक यू सर. चले सर स्टार्ट सब कर दें.
Diversity is a fundamental part of life on Earth. We observe it every day in every aspect of our lives. Variation is also a natural feature of biological systems. Genetic variation has long been associated with complex disease states. In the case of cancer, genetic heterogeneity among cells is the norm, and accurately assessing the genomes of single cells is critical to individualized therapy. Primary template-directed amplification, or PTA, amplifies individual genomes in preparation for downstream genetic analysis in a way that offers unprecedented accuracy and uniformity. Similar to current methods, primers first anneal to the target genome, called the primary template. When DNA polymerase Phi29 binds to the primer and template, it synthesizes a complementary strand from the primary template. Chain-terminating nucleotides are then introduced, restricting amplicon length by disengaging the Phi29 enzyme and preventing further extension of the amplicon. The shorter amplicons have a significantly reduced propensity for reuse, replicating only a handful of times before becoming fully double-stranded. This preferentially directs the primers back towards the primary template of interest. Primary template-directed amplification provides researchers with an unprecedented level of cellular resolution, enabling a new era of genomic discoveries across a multitude of applications. Pta offers superior performance compared to traditional MDA. Redirection of primers to the primary template prevents the propagation of errors that is common with traditional MDA. Pta also offers significantly higher breadth of coverage and uniformity, resulting in increased sensitivity. With this increase in coverage uniformity, researchers observe far less allelic imbalance compared to MDA. Some examples of applications of Pta include cancer diagnosis and treatment, monitoring disease progression and minimal residual disease, single-cell bacterial sequencing, measuring CRISPR off-target rates, pre-implantation genetic testing for in vitro fertilization, and accurate sequencing of low-input samples for ancestry and forensics. For more information about how Pta can enhance your single-cell or low-input genomic analysis, visit bioscribe.com.
Is it clear? So next is the same explanation for, this is the explanation for the same video. And basically Pta reaction products are double-stranded and may be converted into libraries for multiplex sequencing on Illumina. So there could be the libraries for the multiplex sequencing for Illumina and other short read platform without fragmentation. Now in the sequencing, we will not need any fragmentation because there will be small fragments actually. So if you can see, this is the DNA strand, the longer DNA strand, and the hexamers, the primer, the short primers, they will bind here and they will start amplification. Every hexamer will start amplification, they will bind and the amplification will go parallel to each other. And now when the amplicons are formed, the amplicons will release and these amplicons will become double-stranded. After they will become double-stranded, no, not any primer will bind with these the secondary products, the products, because they will probably they will not have any site for these primers because they are short-stranded. In the long strand, there will be multiple sites for these primers. So actually, the terminators were used and they form short amplicons, which could not be used for further amplification. And so that will mean only the amplification will occur from the primary strand, and therefore that is known as primary strand dependent primary transcript amplification. So primary strand dependent amplification. So next is, now you can see many products, they are formed from the same strand, from the primary template. So primary template dependent amplification is this. Okay, so after that, the next one is RPA from Pta. Now we will move towards RPA, and RPA is recombinase polymerase amplification. And for recombinase polymerase amplification, let's watch the video. And this is a bit more complex than the previous one.
Let's talk about recombinase polymerase amplification. What is it exactly? RPA is a type of isothermal polymerase chain reaction, or PCR. How does it work? As in usual PCR, you have forward and reverse primers sequenced to amplify and polymerase. but the difference is our primers are integrated into the helix by a recombinase protein UVS6. Then single-strand binding proteins stabilize, forming single strands. As you can see, recombinase works only in the presence of ATP and crowding agent. It's usually a high molecular PEG. Later stabilizes the recombinase primer complexes. After that, recombinase disassembles, and the strand displacing DNA polymerase binds to a 3' end of the primer to elongate it. Of course, polymerase displaces SSBs, and the cycle repeats over and over. There are some characteristics of RPA. Using this method, you can amplify RNA, small interfering RNA, single-stranded and double-stranded DNA, etc. Since RPA is isothermal, amplification can take place at the constant temperature from 25 to 42 Celsius degrees. Amplification is extremely rapid, about 30 minutes on the average. It can detect one copy of target sequence, so it's very sensitive. Also, you can do multiplexing. Main pros of this method are it needs no special expensive equipment, it's very sensitive and selective, and there is no narrow temperature control as in usual PCR. Main cons are it needs fragments up to 1.5 kilobase pairs, and it's very sensitive to mismatches. What is needed for RPA? Of course, you need strand displacing DNA polymerase that doing elongation. You need target sequence, primers, DNTPs. Of course, you need recombinase protein UVS6 that inserts primers, single-strand binding protein that stabilizes single-stranded DNA, and crowding agent that stabilizes recombinase primer complex. Also, RPA can be carried out on the surface. It's called solid phase RPA. Commonly, one of the primers is attached to the surface. Of course, such a method has some obvious drawbacks, but it's good for specially resolved multiplex amplification when each amplified sequence turns out to be attached to the particular place on the surface. And of course, it allows coupling of amplification and different detection techniques. Thank you for watching. Good luck.
Okay, so this was RPA. Let's take a 5-minute break and we will be coming back. Next of the methods is RCA, that means rolling circle amplification. And for rolling circle amplification, there will be circular template, circular DNA or RNA template, and let's see what the videos say about it. This is actually too much important, and a natural process already you know in the plasmids amplification. Some of the viruses also employ this. The previous one, like recombinant DNA, recombinase plasmid, is actually already, and also that is also a natural process, but we know little about that, and this is more famous and common actually.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.
So this was rolling circle amplification. And next is strand displacement amplification. In this, the strands will be displaced as the name indicates.
Hi, I'm Dr. Seifert with TampaPath.com, and I'm here to explain Strand Displacement Amplification, or SDA, a molecular technique. SDA is an isothermal technique for the detection of DNA or RNA targets. It is isothermal because the target amplification step takes place at one temperature and does not require a thermocycler. SDA's applications include the detection of tuberculosis, chlamydia, gonorrhea, and HIV viral load. While not requiring the expense or maintenance of thermocyclers, one drawback of SDA is that it is less stringent. Primers can non-specifically bind to the template. The process itself uses two special primers. The goal of the target generation phase is to generate a new special version of the DNA target we want to amplify. The reaction mixture contains deoxyuracil, guanine, adenine, and thiolated cytosine. In this example, we will be using a DNA template, and this example shows modification and amplification of only one strand. The amplification primer contains a special restriction enzyme cut site and is specific for a known region of the template DNA. Upstream of this binding site, the second primer, the bumper, binds. Extension with DNA polymerase takes place simultaneously and uses the amplification primer and its strand off of the DNA template. The strand that was displaced in the previous step will be the template during the target amplification phase. The modified strand, which has hybridized to another opposite strand, contains the restriction enzyme site that was present in the amplification primer. This site is recognized by an exonuclease. Normally, exonucleases cut both strands of DNA when a site is recognized. However, since our reaction mixture included thiolated cytosine, the exonuclease only cuts one strand. This is called a nick. DNA polymerase recognizes the nicked site and synthesizes a new strand, starting from that position. This displaces the previous strand. This process of nicking, extension, and displacement occurs over and over again for both DNA strands exponentially. Thank you for watching this presentation on strand displacement amplification.
Oh, and one of the application of strand displacement amplification. So for that, let's check this video.
The sequence specificity of DNA means that the kinetics of DNA strand displacement can be directly simulated using the DSD tool. For example, an elementary strand displacement reaction is shown in which an input strand displaces an output strand. The input strand has the correct complementary nucleic acid sequence to bind to the toehold and once bound, it can displace the output strand base by base. Because single strands are used as both inputs and outputs to this system, the output from one circuit can be used as the input to another, so that scalable circuits can be created. In theory, the rules of strand displacement can be used to program any logic system that can be expressed as a chemical reaction network. For example, we can implement the reaction A plus B goes to C, the biological equivalent of a join gate. The first input strand binds to the complementary toehold, displacing the attached strand. This then reveals a second toehold which corresponds to the second input strand, which is then able to displace the output strand. Only when both input strands are present will the output strand be released. The presence of a further strand which binds to the output molecule and contains a fluorescing molecule allows us to track the progress of the reaction. This join gate reaction can be viewed as an abstracted nucleic acid sequence, and the dynamics of the reactions can be simulated using the DSD tool. There are many concurrent reversible reactions even to describe this simple logic operation. Complex systems can become unmanageable very quickly, so the DSD tool was developed to simulate the resulting networks of reactions. As a proof of principle, we have developed a molecular circuit that implements an algorithm for achieving consensus. Given two populations of molecules, X and Y, the algorithm works out which is in the majority. The algorithm is expressed as three reactions. The two populations of DNA molecules, here shown in red and green, cancel each other out to form an intermediate molecule, shown in orange. The second and third reactions are positive feedback loops, one for X and one for Y. Whichever population is in excess will outcompete the other and eventually convert the intermediate back to itself so that an overall consensus is reached. We have implemented this high-level algorithm in a modular way by considering each reaction as a separate circuit. For example, if we start with this population of DNA strands, the dynamic reactions convert X and Y strands into intermediates and back again. Eventually, the X feedback loop outcompetes the Y feedback loop, and the system converges to be fully X, which was the strand that began in excess. In reality, there are billions of molecules interacting in parallel, and the dynamics of the reaction system ensure that the correct consensus is reached. The reactions involved in the consensus circuit are multi-step operations, and a number of strands and output strands with fluorescing molecules are needed. The full set of reactions are complex, and the dynamics of these reactions are abstracted and simulated in the DSD tool, which handles the modelling of this network of reversible reactions in a manageable form. The logical structure is programmed into the DSD tool, and the set of possible behaviours is generated. We see the functioning of the consensus circuit where X and Y quickly cancel each other out to produce the intermediate B, which is then converted to the majority. The result of this simulation is in agreement with experimental data. Designing such strand displacement systems can be difficult and error-prone. The DSD tool simulates all these reaction steps and enables the creation of complex logic circuits using chemical reaction networks.